Welcome to the deep dive. Today, we're jumping straight into a really critical challenge in pediatric cancer, medulloblastoma, MB for short.
Yeah, it's the most common malignant brain tumor in kids. A really tough diagnosis.
And we're focusing even tighter today on the most aggressive subtype, group 3 medulloblastoma.
That's right. Accounts for maybe a quarter of cases, but they have the worst prognosis. Part of the reason is they're just incredibly metastatic.
Meaning they spread easily.
Exactly. Over half of these kids, unfortunately, already have disseminated disease when they're diagnosed. It's already spread through the brain or spinal cord.
And the standard treatments, heavy chemo, radiation, they can work, but the long-term side effects for survivors are often, devastating.
Absolutely. Decades of challenges.
Yeah.
Which is why the research we're diving into is so important. It's not just about stopping growth generally.
No, this is about finding the sort of master controls, the switches that determine the actual identity, the fate of these aggressive cells.
Our mission today is really to trace this specific molecular circuit, the one that seems to decide if these cells stay primitive and deadly, or if they can be pushed towards differentiating, maybe becoming less harmful.
And the key player, the name that keeps coming up in the sources, is this oncogene called OTX2.
OTX2, orthodentical homeobox 2. It's a transcription factor, meaning it controls other genes, and it's definitely a big deal in this cancer.
Yeah, the data shows it's amplified, like there are extra copies, or it's just highly expressed in, over 80% of group 3 and also group 4 myopes.
Huge numbers. It's clearly driving the tumor. But the core question we need to unpack is how? How does OTX2 manage to keep these cells trapped in that really primitive, undifferentiated state?
Like that aggressive stem cell-like state.
Okay, so the thinking is that group 3 myeloblastomas probably arises from these very early primitive cells, stem cells, or progenitor cells in the developing brain.
And OTX2's job seems to be guarding that primitive identity.
Exactly. It actively stops the genes needed for normal neural differentiation. It essentially tells the cells, nope, don't grow up, stay undifferentiated, stay aggressive.
So how did the researchers figure this out? They looked at group 3 millib cells in the lab, right? These things called tumor spheres.
Yeah, tumor spheres are basically a way to grow the cancer cells in a way that enriches for the stem-like ones. They used a well-known group 3 cell line, D283.
And they knocked out OTX2. Turned it off.
They silenced it, yeah. Then they did this really comprehensive analysis, multiomics, looking at everything. Gene expression, but also the chromatin landscape.
Chromatin, that's the DNA packaging, right?
Yeah.
How genes get turned on or off using chemical tags.
Precisely. Think of it like instruction manuals for the cell. You can put sticky notes on certain pages. Some notes say, read this now. Those are active marks, like H3K4, ME3.
The go signal.
Right. And other notes say, do not read. Those are repressive marks, like H3K27, ME3, the stop signal.
Okay, so they silenced OTX2 and looked at the sticky notes. What changed?
It was dramatic, a massive release of suppression. For genes where the go signal changed, 90% of them showed a gain in that signal. They were switched on.
Wow, 90%.
And even more telling, for the stop signal, H3K27, ME3, 68% of genes where that changed showed a significant loss of that repressive mark. The brakes came off.
So OTX2 isn't just silencing one or two things, it's acting like a broad global repressor.
Totally. It's like a dam holding back a flood of differentiation signals. You remove OTX2, the dam breaks, and genes everywhere start getting activated.
And which genes were most affected? The ones that really sprang to life.
This is key. It was transcription factors needed for making neurons. Specifically, members of the PAX gene family, PAX3 and PAX6, stood out.
PAX3 and PAX6. And how does this connect back to actual patients?
Well, that's where it gets really compelling. The researchers looked at patient tumor data. In the aggressive group 3 patients, the expression levels of PAX3 and PAX6 were significantly lower than in patients with less aggressive types, like WNT or SHH subtypes.
Lower PX means more aggressive tumor.
Exactly. And crucially, the flip side was true, too. Group 3 patients who happened to have higher levels of PX3 or PX6 expression, they had significantly better overall survival.
So it really looks like OTX2 is silencing genes that would normally act like tumor suppressors, like brakes on the cancer's aggressiveness.
That's the hypothesis. OTX2 keeps PX3 and PX6 quiet, and that helps maintain the dangerous primitive state.
Okay, so if OTX2 is the repressor, how does it actually work at the molecular level? How does it physically stick that stop signal onto the PX genes?
Right. This takes us into the epigenetics, the actual mechanism. The first clue was that loss of the stop signal tag, H3K27E3, specifically at the PX3 and PX6 gene locations right after OTX2 was silenced.
Suggesting OTX2 needed to be there to maintain that mark.
Precisely. So they used a technique called chip-piece sequencing. It basically lets you see where a specific protein, like OTX2, is physically binding to the DNA across the whole genome.
And they found it.
They found OTX2 enriched, meaning stuck right onto the DNA at the critical regulatory spots for both PX3 and PX6, specifically near their transcriptional start sites, the starting line for reading the gene.
And you mentioned something interesting earlier, this bivalency pattern at these sites. What's that about? We have the go signal and the stop signal.
Ah, yes, bivalency. It's fascinating. It means both the active go mark, H3K4Me3, and the repressive stop mark, H3K27Me3, are present at the same gene, often at the start site.
So it's both on and off. How does that work?
Think of it as poise repression, like a race car at the starting line. The light is red, that's the repressive H3K27E3, the gene is off, but the engine is absolutely revving, that's the active H3K4Me3. It's silenced but primed for super fast activation if the signal changes.
So OTX2's job is to make sure that red light stays firmly on.
Exactly. Keep it repressed, keep it primitive, but maintain that potential for rapid change perhaps, which might be useful for the tumor later.
Okay, that makes sense. But how do we know OTX2 isn't just nearby? How do they prove it recruits the machinery that puts down that stop signal? That seems like a hard thing to demonstrate.
That's the next crucial piece. OTX2 doesn't place the repressive mark itself. It calls in the specialists. They used co-immunoprecipitation, a technique to see which proteins stick together in the cell.
And OTX2 stuck to.
It stuck directly to EZH2 and SUZ12. These are core components of the polycomb repressive complex 2, or PRC2. PRC2 is the molecular machine whose whole job is to lay down that H3K27Me3 stop signal.
Ah, okay. So OTX2 finds the address, the PX3 and PX6 genes, and then it essentially flags down the PRC2 complex to come over and paint the stop sign there.
Yes, but it's actually even more insidious than that. It's a dual mechanism. They found when they silenced OTX2, not only did OTX2 leave the DNA, but the actual amount of EZH2 and SUZ12 protein in the cell went down too.
Whoa, wait, so OTX2 controls where PRC2 works, and it controls how much PRC2 is even available.
That's what the data suggests. It's directing the complex to the right spots and regulating the overall levels of the repressive machinery itself. It's a really robust way to ensure PAX3 and PX6 stay completely silenced.
That's a really tight grip, a two-pronged attack to lock those cells in the primitive state. Okay, so this naturally leads to the next question, maybe something for you listening to think about. If OTX2 is doing all this to silence PX3 and PX6, what would happen if we just bypassed OTX2 and forced the cells to make PX3 or PX6 anyway? Could we force them to mature?
That's exactly what the researchers tested next with gain-of-function experiments. They took those aggressive group 3 middle B cells and basically forced them to overexpress either PAX3 or PX6.
To see if adding these back could, reverse the cancer phenotype.
Right. And initially, in vitro and cell culture, both PX3 and TX6 looked promising. They both inhibited the ability of the cells to form those primary tumor spheres, which, remember, is a measure of that stem-like self-renewal behavior.
Okay, so far so good. Both seem to act like tumor suppressors initially. They also both reduced levels of SOX2, right, a stem cell marker?
Yes. Both knocked down SOX2 in the primary spheres. But the critical difference emerged when they looked deeper at longer-term effects and in vivo in actual mouse models.
Ah, the real test.
Indeed. Only the cells forced to express PX3 maintained that reduced self-renewal capacity when they tried to form secondary tumor spheres. PX6's effects seemed to wear off.
And in the mice.
Even more stark. Only the mice injected with the PX3 overexpressing cells showed significantly longer survival and had much smaller tumors. PX3 really put the brakes on in vivo.
And what about SOX2 and the tumors from the mice?
Another key difference. PX3 overexpression completely wiped out SOX2 expression in the xenograft tumors. PX6 overexpression didn't have that strong effect on SOX2 in vivo.
Okay, so PX3 is clearly the more potent tumor suppressor in this context. PX6 has some effect, but it falters. This functional divide is really the crux of it now. We need to know why. What is PX3 doing differently?
They dug into the molecular pathways using RNA sequencing to see which genes were turned on or off by PAX3 versus PAX6.
Did they find the unique PX3 pathway?
They did. Both PAX factors affected genes involved in neuronal development, things like GABAergic and glutamatergic signaling, which makes sense for brain tumors. But only PX3 overexpression led to a significant downregulation of genes in one specific crucial pathway, mTORC1 signaling.
mTORC1. Okay, that rings a bell. It's a huge pathway involved in cell growth, metabolism, protein synthesis, often called a master regulator of cell growth.
That's the one. High mTORC1 activity fuels rapid protein production, biomass accumulation, basically everything an aggressive, rapidly dividing, undifferentiated cell needs.
And PX3 specifically turns down mTORC1 activity.
Exactly. They confirm this by looking at downstream markers of mTORC1 activity phosphorylated proteins called PS6 and P4EBP1. Only the PX3 gain-of-function cells showed a clear reduction in these markers. PX6 didn't touch mTORC1 signaling in the same way.
Wow. Okay, so let's trace that circuit then. OTX2 is high, it represses PAX3. Low PX3 means mTORC1 signaling runs wild, unchecked. And that high mTORC1 activity fuels the aggressive stem-like state.
That's the model. It connects the epigenetic control by OTX2 directly to metabolic control via mTORC1, which then dictates the cell's fate.
And was there any final confirmation of this link between mTORC1 and the undifferentiated state?
Yes, they used single-cell RNA sequencing on the tumor spheres. This gives super high-resolution data, cell by cell. It confirmed that the dominant gene program active in these group 3 cells was indeed this undifferentiated progenitor program, full of genes for making ribosomes, translation factors, everything needed for high-protein synthesis.
And that program correlated with mTORC1.
Showed a strong positive correlation with the mTORC1 gene signature. High mTORC1 activity goes hand in hand with that aggressive, undifferentiated state at the single-cell level.
Okay, knowing this whole pathway now, from OTX2 down to PAX3 down to mTORC1, this points directly to a therapeutic strategy, right?
Yeah.
Target mTORC1.
Absolutely. If high mTORC1 activity is critical for maintaining the aggressive state, then inhibiting it should theoretically push the cells towards differentiation and slow down the tumor.
So did they test this with drugs?
They did. They used a compound called PQR620. It's a dual inhibitor, meaning it hits both mTORC1 and a related complex, mTORC2, and importantly for brain tumors.
It crosses the blood-brain barrier.
Exactly. That's a major hurdle for brain tumor drugs, but PQR620 gets into the brain.
Man, and the results. Did blocking mTORC1 work as well as boosting PAX3 did?
The results were really quite dramatic. In the mouse models with orthotopic xenografts, meaning tumors growing in the correct brain location treatment with PQR620, led to a significant increase in survival and a major reduction in tumor growth.
That's fantastic. Did it also phenocopy the PAX3 effects at the molecular level, like reducing SOX2?
It did. PQR620 treatment mirrored what they saw with PACUC3 overexpression. It decreased the stem cell marker SOX2, and crucially, it increased levels of tubulin,
Which is a marker for it.
neuronal differentiation. So inhibiting mTORC1 seemed to force the tumor cells to differentiate, to mature, just like expressing PAX3 did. It pushed them off that aggressive, primitive path.
This is really major progress, then. This deep dive has uncovered this really elegant OTX2-PAX3 gene network.
Yeah, showing how OTX2 uses chromatin modification, those stop signals, to repress PAX3.
Which in turn unleashes mTORC1 signaling, fueling the aggressive, undifferentiated state.
Right. And understanding that whole chain provides a clear molecular rationale for why targeting MTR signaling could be effective against these really resistant stem-like cells in group 3 milli.
It really highlights how digging into these complex molecular controls is absolutely essential if we want to develop smarter, more targeted, and hopefully less toxic therapies for kids with these devastating cancers.
Absolutely. It's about hitting the specific vulnerabilities dictated by the cell's fate program, not just general growth pathways.
Okay, so to leave you, our listener, with something to think about based on the source material, there's a fascinating complexity that remains.
We've talked about group 3 being the most aggressive, but there's another subtype, the WNT subgroup, which actually has the best prognosis, almost 100% survival, typically.
Oh.
Here's the kicker. Both OTX2, the oncogene we've been discussing, and PAX3, the tumor suppressor in group 3, are found to be highly expressed in those low-risk WNT tumors.
Wait, what? OTX2 is high and PX3 is high in the least aggressive type. How does that work?
Exactly. If OTX2 is driving aggressiveness and PX3 is suppressing it in group 3, what on earth is going on in the WNT subgroup? There must be other dominant molecular players, other pathways active in those cells that completely neutralize OTX2's oncogenic drive, despite its high expression.
So the context is everything. It's not just about single genes.
Precisely. It points to a whole other layer of network interactions that determines the final outcome. And figuring that out Well, that's the next challenge for researchers.
A fascinating puzzle indeed. Thank you for joining us for this deep dive into the complex biology of medulloblastoma.